Dear student, I am Dr. Mohammad Nasser Malik. I teach you lignin and flavonoids. This is the disclaimer. All the content material provided here is only for the teaching purpose. And in this, you study lignans and flavonoids. A student will learn introduction of lignans, biosynthesis of lignans, flavonoid, classification of flavonoids. Introduction of lignans. The term lignin was first introduced by Howard in 1948 to describe a group of dimeric phenyl propanoids where two phenyl propanoids molecule are attached by the central carbon atom C8. Lignan are the subgroup of non-flavonoids, polyphenols. You see here, they are widely distributed in the plant kingdom, being present in the more than 55 plant family, where they act as an antioxidant and defense molecule against the pathogenic fungi and bacteria. Following the structure, they contain phenylpropanoids. A is the structure of phenylpropanoids and B is the lignins. Okay. So chemical structure of lignins. Their basic chemical structure consists of two phenyl propanes units like linked with CC bond between the central atom of their respective side chain position 8 or beta are also beta-beta bonds. In their case of the diamonds are called neolignins. Hence their chemical structure is referred to act as C6 to C3 and they are included phenyl propanoids group as well as their precursor of the hydroxycinnamic acids. Lignin can be found in more than 60 family of vascular plants and have been isolated in different plant parts extruded from their resins. Biological activity of lignins are antiviral, anti-cancer, cancer prevention, anti-inflammatory, antimicrobial, antioxidant, immunosuppressive, hepatoprotective, osteoporosis prevention. Based on their carbon skeleton cyclization pattern and the way in which the oxygen is incorporated in the molecule skeleton, they can be divided into 8 subgroups. The main subclass of the lignins is furofuron, furan, dibenzylamine, dibenzyl butylactol, dibenzyl butyrolactones, arylethylene, arylethylene, arylethylene and dibenzocyclooctadienes. Among these groups, the furan uh, and dibenzoyl can be further classified as a lignin with C9 oxygen and lignin without their oxygens. These are the structure of for furan and dibenzyl butanes. They are not present in the free form in the nature but linked to other molecules, mainly glycosylated derivatives. Among the most common lignins, cyclosuccinol, the most abundant one, lyric, succinol, succinol, metrylsuccinol, and 7-hydroxy metrylsuccinol are found. Protophyllotoxin, a naturally acting arylethylene lignins, is one of the most important compound due to the high toxicity and current use as a local antiviral agent. Protophyllotoxins is obtained from the plant of the genus Podophyllum, family Berberissae. It is a mitotic toxic whose derivative have been used as a chemotherapeutic agent used in food rich lignans. The richest dietary source is flaxseed, that is called also linseed, uh, contain mainly of succinols, but also lacri-succinol, pino-resorcinol, and metrorcinol is a good quality. They are the same and same food grains. Okay. Biosynthesis of lignins. The pathway starts from 3 to 4 most commonly hydrocinamic acid, p-coumaric acid, synapic acid, and ferulic acid. Uh, Caffeic acid not precursor of the subgroup of the polyphenol. They arise from the shikimic acid pathway via phenylalanine. The first three reactions reduce the carboxylic groups of the hydroxycinnamates to alcohol group. The formation of the corresponding alcohol called monolignol. It is a p-coumaric alcohol, synapyl alcohol, and porphyryl alcohol. They molecule also enter the pathway of the lignin biosynthesis. The first step which leads in the activation of the hydroxylamine acid is catalyzed by the hydroxylamate coenzyme A ligase, commonly called as p-coumarate coenzyme A ligase, which with formation of the corresponding hydroxylamate coenzyme A, namely ferrolyl coenzyme A, p-coumarate coenzyme A, and synapyl coenzyme A. The second step is NADPH dependent cinnamyl hydroxylase, also called cinnamyl coenzyme A reductase catalase the formation of corresponding aldehyde. In the last step of base depend upon the cinnamyl alcohol hydrogenase, also called as monolyginol dehydrogenase, catalyze the reduction of the aldehyde group to an alcohol group with the formation of aforementioned monolyginols. The first step of dimerization of monolyginols involves in the intervention of stereoselective mechanism, or more precisely enantioselective mechanism. Plant lignans exist in the positive or negative enantiomers. Hydroxylamic acid is hydroxylamate are also less of aromatic acid or phenyl propanide having C6H2C3 skeleton. These compound are the hydroxy derivative of cinnamic acid. These are the structure of the different types of lignans: synaptic acid, synapyl coenzyme A, synapaldehyde, synapyl coenzyme, and further lignin forms. Ferulic acid, ferulic coenzyme A, coniferaldehyde, coniferaldehyde alcohol, phenolcerol, lancinolone, succinolone, metrinoxol. P-coumaric acid, P-coumaryl coenzyme A, P-coumaraldehyde E, P-coumaryl alcohol A, and lignans. Finally, all step lignan is formed. Flavonoids. As you know, flavonoids are the most abundant in polyphenol in human diet. Over 4,000 have been identified. Latin word flavus meaning yellow. They are responsible for the colors, flowers, fruits and leaves, protection and ultraviolet radiation, pathogenesis, herbivorous. In fruit and veg also used in the form of glycoside and sometimes as a glycoside characteristic of flavonols. Chemically, flavonoids have general structure of 15 carbon skeleton which consist of 2 phenyl rings A and B and heterocyclic ring. This is the flavin nucleus. Classification of flavonoid, they can be divided into different subgroups depending on the carbon ring on the which beta ring is attached. There are and substitution of carbon ring flavonoids in which beta ring is in position 3 to 5 rings. Three are the called isoflavonols. Those in which beta ring is linked position neoflavonols. While those in the which beta ring is position 2 can be further divided into several subgroups on the basis of structural features. Carbon rings, they subgroup are there: flavonols, catechin, flavanols, and anthocyanins. Finally, flavonoids have with open carbon rings called chalcones. Xanthone like flavonols. Flavonols are mainly found in the cereals and herbs. Butanol, epigenin, tangerine. Flavonol are 3-hydroxyflavanol. Food rich in flavonols are the human diet like fruits, vegetable, beverages such as red wine and tea. Quercetin, camphorol, myristicin, fusticin, glycine, isomamidine, pecrolamine, rizampin, and pyronoflavonols and sulfuranols. These are the examples. This is the structures of flavonols. Flavonones. This type of flavonoids are very aromatic and colorless ketones derived from the flavanol that are often occur in the plant of glycoside and flavonols. Hesperidine, mariginin, and irodine and hemodictyline. Flavonols classified flavonols 3-hydroxy, 2-hydroxy, 2-phenyl, chromone, and 4,1. Name is backbone. Flavanol are 3-hydroxy or 2,3-dihydroxy taxipholline and dihydroxy quercetin and dihydroxycamphophyrol. Flavanols, the flavans and the benzophenone derivatives used as a 2-phenyl 3,4-dihydroxy dihydro 2,4 chromonoskeleton include phenolphthalein. Example of the catechin: galactosine, catechin 3-galactate, galactocatechins, galactase, and epigalactogene. Flavan 4-ol, 3-flavon, 3,4-diols. These are the flavon 3-ols, flavon 4-ols, flavons, and their structures are here. Anthocyanins Cyanidin. Anthocyanidin are the egg-like cone of anthocyanins. Anthocyanin are the very large group of red-blue plant pigment. Anthocyanin occur in the all higher plants, mostly in the flower, fruits, but also in the leaves, stem, roots. They are very good antioxidant. Health benefit of the flavonoid: they are regular consumption of associated with reduced risk for number of chronic disease, include cancer, cardiovascular disease, neurodegenerative disease. They are good antioxidant, anti-inflammatory agent. These are the references of the this lecture. So you go through it, and if any queries, any question, you ask me.